All right, Mark, 3, 2, 1, hit it.
Hello, everyone, and welcome to the Explorers Club Monday Night Lectures. I'm Mark Fowler. I'm the Vice President of Conservation at the Explorers Club. And tonight I am super psyched. We have this crazy evening where we're focusing on snakes, snake bites, and venom, and antivenom, which is what the really exciting work we'll be talking about tonight. As many of you know, globally, snakes are some of the most hated and revered animals on the planet. I personally, as a person who's a wildlife lecturer, I love snakes, and I've worked with snakes my whole life, and I find them to be some of the most unique living animals on the earth, on our planet. Whether they're poisonous or constrictors, not poisonous, excuse me, venomous. Thank you. That's a mistake we all make, but whether they're venomous or whether they are or whether they're constrictors, I mean, they're some of the most unique animals on earth. So I've grown up with them. I've had, a 9-foot boa bite me and wrap me, and that was sort of fun and interesting. But tonight we're going to be talking about the incredible world of venomous snakes. Venomous snakes are killed. Everybody kills them the second they see them, or they think almost all snakes are venomous, and they're terrified. And the truth is, as animals, that we have an instinctual fear of being bitten, scratched, or mauled. Okay, so many of us are terrified of animals, especially something like a snake or a potential venomous snake. But here's the truth. If someone's bitten by a snake, it's never the animal's fault. It's never the snake's fault. These animals, they exist in a different world. They exist in a world where we are not a prey species to snakes. If we ever get into their habitat and somehow scare them or touch them or step on them, that's when people get bitten. So we have to understand this world, and we have to understand the risks. According to Bill Gates and to many more, they say it's one of the most deadly of our wild animals. But the people that we have on tonight, I have to tell you, I not only love snakes, and I know the people we have on tonight also love snakes, because they're working on bridging this gap between bites and people who end up the mortality of snake bites. And they're working with antivenom and all kinds of new techniques to save people's lives so that we don't have to have this sort of love-hate with snakes. So I'm very excited. We have one of the most accomplished groups I've ever had on one of these Monday night lectures. There are more doctors on this call than I've ever had on an Explorers Club evening. So tonight we have 4 speakers. We have Jordan Benjamin. He is this amazing herpetologist, wilderness paramedic. We've got Dr. Kate Jackson, who's a professor of biology at Whitman College. And I'll tell you something, she has done the most amazing work in the Congo and has inspired many of the people in this field, many of the people on this call. We also have Dr. Nick Brandehoff, who's as well going to be joining us and speaking. He's working on some of the most cutting edge work with antivenom. And we also have one of our fellows at the Explorers Club, Dr. Matt Lewin. And Matt's working on some very novel and new techniques to address the snake venom and snake bite issue from another angle. So tonight's going to be a great evening. I'm just excited to learn a lot, and I'm very excited to share our first speaker with you. Jordan is top-notch snake expert and anti-venom expert. He runs the Asclepius Snakebite Foundation. And we are very honored to have Jordan. Jordan, if you can share a little about yourself and then give us a talk about what you're working on. And I know that that you work with Nick and you work with Dr. Kate Jackson. So if you can tell us a little bit about what you're working on, and then tell us the challenges that you and your team are facing when you're addressing snakebite and antivenom, et cetera, so that we can take this tropical disease and actually make it less of one, because I know survival rates in the United States are much higher than the rest of the world. And I know that you all are working to try to reduce that challenge and make it survivable in the other parts of the developing world. So without further ado, Jordan from the Asclepius Snakebite Foundation. Jordan, please join us.
Awesome. Thank you so much. It's a privilege to be here. tonight. Can you guys hear me okay?
Yep.
Everybody hear me? Okay, awesome. I'm going to screen share this here. So my name is Jordan. I am the founder of the Asclepius Snakebite Foundation. I'm a herpetologist and snakebite researcher, no conflicts of interest. I have worked in Africa, working on the issue of snakebite for about 12, 13 years now. Worked very closely with Dr. Brandehoff and Dr. Jackson, good friends with Dr. Lewin. So it's a small community here. What we try to do as an organization is to essentially bridge the gap in knowledge between the different disciplines that snakebite sits between. And that's going to be sort of my goal in the opening part of this talk, is to convey the public health challenges facing clinicians on this side, but also to talk about where this intersects. Because we have a group tonight that really reflects, I think, all three of the critical areas for managing snakebite, which is essentially the clinical research, the clinical management, and then of course the snakes and the conservation. So you can break snakebite down loosely into snake factors and human factors. And when we're thinking about what determines the severity of envenomation, we sort of break it down there initially. So on the snake side, we have the species snake, we have the quantity of venom, we have the composition. And on the human side, we have a whole lot of other factors, like the health of the person, what was the healthcare seeking behavior, how did they respond to that actual bite, what was the treatment delay, what treatments are available. So we'll come back and orient to this figure here sort of throughout the talk. Just remember, we've got a circle on the left of sort of human factors, circle on the right of our snake factors, and then right in the middle is where we come together. So I think one of the things that surprises people about snakebite is just the scale of this issue. We're looking at 5.4 million bites every year worldwide. About half of those result in envenomations. So that's a bite where venom is injected, as opposed to a dry bite, which we'll talk about. There's about 138,000 deaths worldwide every single year, and probably well over 500,000 disabilities. This sounds like staggering numbers, but in reality, most experts feel that this is probably 3 to 5 times less than the actual morbidity and mortality. And if you look at this map here on the right, I think it does a good job of explaining this. Much of Central Africa, for instance, most of Africa in general, we have very limited knowledge. And many of these countries are places where, if you look at the distribution of snakes, they sit along the equator, venomous snakes, and they're certainly spread throughout all over the world, but there is a concentration right in these areas where we would expect to see a significant amount of bites, and where it's complicated by issues of distance to healthcare reporting systems, and a number of other factors that play into that. So these numbers look staggering, but they are also probably significantly smaller than what we're looking at. If we look at the actual issue of snakebite, so we looked at the numbers overall. If we dive down and look at this from a sort of a person perspective, the impact of snakebite is really disproportionately borne by victims living below the poverty line, but it's also a disease process that tends to impact families over generations. Most of these areas, people are reliant on subsistence agriculture, excuse me. So you have people like this gentleman here who have to go out and work in the fields, and they're using hand tools. And most of these bites are going to be either lower extremity, so foot, lower leg, or hand is the remaining portion there. And most of the people who are bitten are going to be the folks that are performing this labor. So it's about 70% men, it's about 20 to 40% children, and it's individuals who function as the breadwinner for their family. And when they receive these bites, if they don't get treatment in a timely manner, they're likely to lose that arm or that leg, which puts them out of work for the rest of their life, if they're lucky enough to live. And that has a massive effect on the village when you look at the cost of treatment, and then the cost of that lost productivity and lost ability to contribute to running their own household and supporting their village. Most of these bites happen in rural areas. So worldwide, about 95% of envenomations are going to occur far from medical care. And less than 10% will receive actual antivenom, which is the definitive treatment for a snake bite. As far as the actual snakes here, so most snakes are harmless. We have about 3,900 species, just under 4,000, of which we could classify 700 as venomous. And that's sort of a textbook venomous. It means they have some venom and some capability of delivering it. So they have some medical significance, but it could be something like the African garter snakes, for instance, which cause nasal congestion and headache. It could be, any number of bizarre manifestations that we don't believe have a lethal risk to people at this time, but sometimes they get upgraded to that list of 200 potentially lethal snakes. So out of 3,900, 200 we know have venoms that could potentially kill you. Of those, about 25 to 50 are going to be responsible for most deaths. And the key concept for this to dive into is that it's not just about this concept of LD50, which is the lethal dose, sort of drop for drop, how potent is that venom? What it's really about is what is the frequency of that human-snake conflict? Because all of these snakes here are very, very large venom yield, very potent venoms, but they don't come into contact with people very often in a snakebite situation. Taipan does in Papua New Guinea. In Australia, they really don't. Tree cobras, very rare, difficult to find. Bushmasters are absolutely huge, but again, very rare that they actually bite people. So drop for drop is not really the best way to measure it. What we sort of need to look at is what's the actual number of bites, deaths, and disabilities. And we'll see a pretty clear disparity in how these play out in terms of the availability of medical care, and then also the sort of pressure on those snake populations and human populations to mash up together. So Oceania is a really interesting area because we have about 1.5 thousand envenomations annually and about 500 deaths. Now, virtually every snake in this area, every venomous snake is very venomous. The elapids are quite dangerous snakes, but over here you have a large area of land. You don't have people that are running into snakes very often. And when they do, Australia has one of the best aeromedical retrieval systems in the world. So you can have a plane come out, bring you antivenom, intubate you, and bring you to a hospital and put you on a ventilator. That's generally going to be sufficient to save a snakebite patient with a neurotoxic bite in a modern medical facility. Then we also have Papua New Guinea, which is sort of the other side of the coin there, where most of those 500 deaths are occurring in Papua New Guinea, where people are coming into contact with snakes in the forest. Medical care is limited, and antivenom availability, although it's improved significantly, is still less available than it might be to patients in Australia. So if we look at Asia, we'll just continue this sort of analysis, right? We have a lot of deadly snakes. We have variable medical care. We have very frequent human-snake conflict because of the population size, the areas they're expanding into. This is probably pretty good data. India in particular has done some very significant detailed studies on snakebites. I think that this data here is probably what we would see in places like Africa if we were able to get better data. So we may see that this number triples, and it probably will. So in Africa, we think there's 500,000 envenomations and 30 to 50,000 deaths. So we're all sort of settling on 50 as a minimum. And here we have many deadly snakes. We have poor access to medical care in the vast majority of the continent. And then we have very frequent human-snake conflict. So all of these rural areas where you're far from medical care is where these bites are happening. And then in Latin America, this is an area where, to be honest, I think we probably need some more data as well. They don't have quite the rapid lethality with some of their species that we would see in Africa and Asia. They don't have their cobras and kraits and mambas, but they do have a lot of vipers that cause disabilities, and they do have coral snakes, which can cause paralysis. But we're looking at probably 60,000 envenomations. I think this number is probably lower than the actual number, and probably the Amazon would account for some of that and the lack of data there. This doesn't quite add up based on what we're seeing. But again, this is more of an Asia type situation, just with snakes that are a little less likely to kill you as quickly. And then we have Europe and the US. So in Europe, 5,000 envenomations, 5 deaths. In the US, 10,000 envenomations, 5 deaths. The last thing I want to touch on here is this idea of a vicious cycle of snakebite, because this is really what drives the problem from a public health standpoint. And also what really needs to drive all of our interventions from a herpetology standpoint, from a venomic standpoint, from a public health standpoint, really all of those sort of cross-disciplinary areas. So what we see in Africa in particular, but also in Asia and South America, is that when a person is bitten by a snake, there's a certain amount of mysticism that is associated with that. And traditional healers play a large role in the initial treatment of those patients due to both price and cultural beliefs. So generally speaking, a patient will be bitten by a snake and go to the traditional healer first. And here we're wasting, we have time, we're burning time here, and time is tissue with a snake bite. Once they arrive there, they will typically be prescribed some concoction of dried snakes and dung or herbs here. This gentleman gave some patients in Benin a concoction of herbs and snakes, and essentially said, take it for a couple of days, and if you don't get better, go to the hospital. So we've got more time that's sort of ticking by here, right? More tissue damage. Part of the challenge here is that we've talked about our snakes. We have about 200 that could kill you. There's a lot more that won't, or potentially won't cause any harm. So some portion of snake bites, if everyone who's bit goes to the healer first, a significant portion, probably 75%, are going to get better because they were never actually sick. And even with those snakes that we know are dangerous, like this rhinoceros viper here, a venomous snake about 25% of the time won't inject venom because it doesn't want to waste that on something that's not a prey item. It's metabolically expensive to make. So 25% of the time, your known venomous snake, I know it's a cobra and it bit me, but I went to the healer and I got better. Once again, they got better because they weren't sick, but it really reinforces this idea of going to the healer who's culturally more comfortable as an initial destination, who is significantly more affordable, and seems to have a pretty good outcome. Unfortunately, what this means is that all the patients who are actually sick get a lot sicker by the time they make it to a hospital. So they have to deal with transportation issues. In Guinea, a lot of our patients take 18 hours or more to reach a clinic. When they finally arrive at a clinic, there's variable quality of medication, of antivenom, of training on snakebite. So they may get there and not find any antivenom. They may get there and find that there's fake antivenom and purchase that at full price, and then the person doesn't get better, which further reinforces this idea of going up to the healer. And ultimately, if they stay alive, they may be referred elsewhere, further down the line, finally receiving antivenom here. But by this point, a lot of damage has been done. And we can save a significant number of snake bites, even very late with good antivenom. But there's going to be a lot of complications with those cases in many situations. So ultimately, what we have here is a circle where people are essentially given the illusion that the healer is the one that saves them from snake bites. And in reality, by going this route, they're really reinforcing this cycle of bad outcomes and pushing patients away from the modern medical system, or at least away from antivenom, which is critical to treatment. So we need to sort of think about all of our interventions in the context of both these two circles, the snakes and the snake bite, and all of the different disciplines here, and then also trying to target various aspects of that cycle. So with that, I think I will hand over to Dr. Kate Jackson, talk a little bit about the snakes and the pressures on them in a wild environment.
Hey,
Jordan, what that means for people. Yes.
Thank you, Jordan. Wow. Talk about eye-opening. I don't think any of us realize the level of the challenge with snake bites, or I don't think we realize that, it's actually based on the area and the access to healthcare and the poverty in the area. I mean, I couldn't believe the amount of bites in the United States and only 5 people die per year. And then you look at these other regions and it's just the opposite. This is really important work. So I'm super grateful. And so but I'm very honored to be introducing Dr. Kate Jackson. She's the professor of biology at Whitman College in Walla Walla, Washington. She teaches courses on anatomy, herpetology, vertebrate evolution, and zoology. And her research team explore morphology, biodiversity, and evolution of amphibians and reptiles. She's worked all over the world, but primarily much of her field work has been in the Republic of Congo. And she has large teams of students that go out there and do groundbreaking work. And she also has written multiple books. So I'm just excited to hear from Kate, Dr. Kate Jackson. And I'm really excited because Kate has an understanding of both worlds, the conservation. She works on the conservation side and the snakebite anti-venom work, and she's passionate about both. So, you know, Kate, we've seen how snakes are some of the most feared animals on the planet. I mean, in the Bible, for God's sake, the enemy is the snake, you know, and you find this around the globe. You find this sort of evil, this evil image of a snake, but in reality, snakes don't intend to bite people, and we aren't in their prey base. And so I'd love to hear your take on the conservation as well when we hear about your work. So without further ado, I'm very excited to introduce Dr. Kate Jackson.
Thanks very much, Mark, for that introduction, and thank you, Jordan. Mark, I have just one thing to add to your introduction, and that is, I would argue people are not afraid of snakes. People are afraid of the unknown. And you never meet a person who's knowledgeable about snakes who's afraid of them.
Good call.
People are afraid of the unknown. My young child, when he's falling asleep at night, says, "Mommy, I'm scared." And I say, "What are you scared of?" And he says, "I'm scared from the unknown." And you can't argue with that, can you? So I would like to share my screen now. I'm going to share my screen with you. And what I want you to see here is one point that I will make above anything else. And that is, when it comes to snakebite, the snakes are not the problem. So snakes are an important part of functioning ecosystems on the planet. And I'm going to explain to you how snakes are not the problem in a couple of maps here. So here is a map, and this is part of an effort to estimate what are global snakebite hotspots indirectly. So this is showing just one variable on this heat map, is how many World Health Organization category 1 and 2 snake species are present in places. And you'll see that there's a lot, there's my field site in the Republic of Congo, lots of red and orange there, but also in other places, see, in Southeast Asia, Australia. So this is where we have category 1 and 2 venomous snakes. Okay, the next map I'm going to show you is slightly different because we're adding one feature. So this is, where are there category 1 and 2 venomous snakes, and also no effective treatment is available. So what you see here is, like what Jordan was saying about Australia, that if you have effective treatment available, then people don't die from the snake bites. And so even in, you know, parts of Southern Africa where snakebite treatment is available, we don't have the problem that we have right here, where no treatment is available, no data are collected on snake bites. So this is our starting point. And I'm going to try to take you to show you some places in the Republic of Congo where I and my research team have been working, and give you 2 different examples of very different types of habitats where human-snake conflict is occurring in different ways. Okay, so the first one I'm going to talk to you about is about a mining site. And this is a place where humans and snakes are in conflict because there is a mine planned. And this was an area, let's see. So I and my team were contracted to do a biodiversity survey of amphibians and reptiles. So here we are doing this in 2010. And here is the location on the border, so in Congo, but right near the border with Gabon. And this is an area where they were preparing to strip mine or surface mine for iron ore. And the place that they're going to surface mine is 3,000 dense square kilometers of dense primary forest. And the mining company that's going to do this is going to borrow money from an international monetary organization. And in order to do that, they have to show that they're doing this right, environment-wise. So they are commissioning a biodiversity study. We are the herpetology team. They also had people studying other taxonomic groups. And they had us all in doing rapid assessments of various sites within this area. And then as a mitigation measure, they are going to pay to buy an area of comparable habitat nearby that will be protected as a national park. And so to make sure it's going to be an equally comparable type of habitat, we have to document what's there. But it was a kind of a weird place to be a herpetologist because you feel you're like a paleontologist. You're studying things that are there but are going to be gone because it's the whole habitat's going to be surface mined. So here we are. Here's some of the team. This is, I point out especially Ange Zassibulu, who at this point was a graduate student, but you'll see him later on. He's now has completed his PhD. And here we were taking tissue samples, collecting, surveying amphibians and reptiles broadly. So here is a photograph of the site, dense primary forest, some forest savanna mosaic. That's what this site is like. And here's some more of that. That's the Ogooué River near Gabon. And lovely swamp habitat. If you're a herpetologist, you like this kind of thing. And then among all these things, we find lots of snakes, and including things that are exciting for herpetologists. I could talk a long time about the different snakes. We also are, I remind you, doing herpetology in the broadest sense. So we're collecting things like this bush snake, but at the same time, things like this monitor lizard, like this Conyxis, African hinge-back tortoise that's got a carapace on a hinge that it can open and close to protect itself completely, and many amphibians. We're testing the amphibians for chytrid fungus. We're doing real herpetological biodiversity stuff at this site. And some of the things that we find are herpetologically exciting. So this is just one little thing that's a real highlight for a herpetologist. Here is a snake that we came across on our way to check our pitfall trap line. And checking our pitfall trap line, here's this snake with white spots on its lips. And this is Bothrolichus ater. And we can identify this from scale counts. It's a kind of surprising snake in many ways. It's called the laurial-pitted snake sometimes because it has this concavity between the eye and the nostril, which is reminiscent of what one sees in pit vipers, who of course have heat sensory capacity. But it's not a viper even. It's a lampriphyid snake, and a harmless one at that. So we don't know anything about the biology of this pit structure. Somebody looked at one in a museum collection and looked at stomach contents as evidence maybe that could support whether it even preys on warm-blooded things, as you would expect for a thing that has a thermosensory pit. So we're looking at that level of very little being known about it. If you're a herpetologist, you'll be excited by this. If you're not, you probably won't. But the number of dorsal scale rows is sexually dimorphic in this species. So very unusual. 17 scale rows in males and 19 in females. And this photograph is sort of special because it's the first time this species has been photographed alive, ever. So the last time, there are some specimens in European collections, but the last time one of these was collected predates cameras that one could easily bring into the field. So to give you a sense of the level of rarity. So this is stuff we're collecting in a place that's about to be strip mined. Okay, so that as this is just the area being cleared for the airstrip, but you see the orange dirt, that's iron ore. The next site I'm going to take you to is a very different one. So we had on the one hand at the Zanaga mine site this very pristine, perfect, undisturbed until now habitat. On the other hand, we have this other site. I'm taking you to the biggest city in the Republic of Congo. Okay, this is Brazzaville, a city of a couple of million people here on the border between the Republic of Congo and the Democratic Republic of Congo, much larger country on the other side of the Congo River. And this is an aerial view of the city of Brazzaville. And right in the middle of this city of a couple of million people, there are some areas of forest fragment, heavily anthropologically disturbed areas. But looking down on this, you can see during the colonial period, the French laid out boulevards and had palaces and palace grounds and so on for government agencies of various kinds. And it happens that this is now one of the best studied sites in all of Central Africa. The reason being that right here in this area here, this is the City of Science. It's the part of the university campus where all the science research labs are, including my herpetology team. So here is the City of Science forest in the Pat d'Oie Forest Reserve. And here is Dr. Ange Zassibulu, who's a herpetologist here, my former PhD student, and my current PhD student, Lise Mavungu. And so these are, this is one of the most well-studied sites for snakes because the herpetology lab is right here in this building. And much as Ange would have loved to be able to go and do field work somewhere as cool as the Zanaga mine site, somewhere pristine, somewhere undisturbed, that costs money to launch expeditions of this kind. And funds he didn't have available, so he could have sat around and waited, or he could do herpetology where he is. And of course, he chose to study the snakes he can study right where he is. And it turns out that right in the middle of a major urban center on basically a university campus, there are snakes to be found. So these are just forest fragments. Here is the abandoned zoo. So much has been, this was a zoo prior to the War of 1997, and now it's sort of the wreckage of a zoo. And we have these fragments of forest associated with what are university buildings of various kinds. We have little places where water flows, and this doesn't look like much. But people wash their dishes and their clothes there. And, you know, just tell you a bit about where we're going here. Ange found a water cobra there, you know, so these are snakes coexisting with a very dense population of humans. So, whereas on the one hand, Ange would have preferred to go somewhere more exciting, this is perhaps the most relevant study he could possibly have done for humans interested in human-snake conflict. Because, you know, heavily disturbed habitats, exposed to pressures from human population growth, those are increasingly reflect the reality in tropical Africa. We're going to see more of this type of snake habitat, not less. So this survey takes on a new kind of importance because this is a place, a city where records are not kept for snakebite. Snakebite treatment is not available. And yet you have people, and here's this children's playground where we're collecting snakes. And you want to know what kinds of snakes they are? All right, then. So here we go. So here's a big, long species list, and I'm going to explain some of those. But the first thing I want you to see on this chart is that the numbers aren't all even. So this is something we often see in snake populations in really disturbed habitats like these. Disturbed habitats, we have the species that can survive or surviving, but some are really just doing great. So there's very uneven numbers of snakes. So in a collection of, say, 100 snakes or so, you find that almost a third of them are of one species. And, you know, a couple of species are really dominating what there are. So let's have a look at what are these snakes that are coexisting with humans. And especially let's look at some of the venomous ones. So I want to point out there's a forest cobra. So forest cobras, you know, right on a middle of an urban center like that, that's a World Health Organization Category 1 species. We also have some green mambas. So Jameson's mamba. These are also World Health Organization Category 1 venomous snake. Okay, what about that one that there's so many of? So the most abundant snake of all that just really seems to thrive in Brazzaville are night adders. These little night vipers in the genus Causus. So these are a category 2 venomous snake. So these are not among the most lethal venomous snakes, but they can certainly cause a painful bite and terrible tissue damage and things. So things that would require medical treatment. And then finally, this is the water cobra here that I mentioned that Ange found in that place where people are washing their clothes and things. So this is a water cobra, and the picture is actually of one I caught in a different part. I caught this one in northern Congo, and it's nearly 2 meters long. You know, snakes to have coexisting with people. This is interesting. This is a World Health Organization Category 5 venomous snake. So what if a Category 1 is very serious? What is a Category 5? A Category 5 is a venomous snake where, you know, it might well be a medically important snake, but they can't really say for sure because data are not, there are no data on it. Now, the reason here is that whereas these two are fairly broadly distributed types of venomous snake, and there are data from other countries where they occur, the water cobra is restricted to the forests of Central Africa. And in none of those places are thorough records kept. So we are kind of underestimating the potential for danger from these, because we're having to extrapolate a lot of what we are expecting for Central Africa just based on the species that exist there that are broadly distributed ones. So I wanted to mention that as a thing. And I think I'm probably out of time by now. But I want to mention that here in Brazzaville, at the herpetology lab, Lise and Ange, as herpetologists, are often called on by people who have been bitten by a snake or there's a snake. So here was a day when we're told that outside the physics building, the groundskeeper has found a snake. So we go rushing over there with big snake pincers and stuff. And that time it was just a little thread snake, Leptotyphlops. So that was, but people don't know how to tell snakes apart there. No snakebite treatment is available. One time, Ange was called because a student had been bitten by a snake while filling her water bottle at a water pump outside the college dormitories. And she had been taken to the hospital, and somebody had managed to kill the snake and save it. And eventually, 3 hours later, they got Ange to come to the hospital because the hospital doesn't really know how to treat snake bites. They are not equipped for that. So by the time Ange got there, the student was on an IV drip, and they were giving her antibiotics and a rabies shot because it was an animal bite. And luckily, you know, Ange is a herpetologist, he's not a doctor, but he was able to say, okay, this is a house snake. It is a harmless snake. You're going to be all right. And luckily, at least he was able to deliver good news there. Sometimes he's not. So a mining operation contacted Ange because one of their employees had been bitten by a snake, and it was a place where Ange had done a survey. And so he, based on their description and then on the picture of the snake, was able to say, I'm afraid that that is a stiletto snake. I saw a lot of them there. I told people not to mess with them. And he's going to die if he doesn't get medical treatment probably. And no medical treatment is available in Congo. They tried to medevac the guy to South Africa for treatment, and unfortunately he died. So at this point, I and my team in Congo are only able to provide information about snakes and snakebite, not any treatment available. And that's something we're hoping to work on with the Asclepias Snakebite Foundation. So I will stop there, and I'll stop sharing my screen and hand this over to Nick.
Hey, thank you, Kate, Dr. Kate Jackson, everyone. Thank you so much, Kate. I mean, what incredible work. And I love that your colleague and your team, your colleague Ange and your team have been studying right in the center of Brazzaville, which is such a unique city in itself. But you're so right that maybe it is the most important study because it's right where all those people, millions of people are surrounding that one area. And you still find that incredible biodiversity of beautiful and incredible snakes. And it's only in those areas where there, like you were saying, there's no treatment, there's little known, that's where the real morbidity happens. And I thank you for saying, I mean, people are afraid of the unknown. It's true. I've always wondered that. Is there an instinctual fear, or is it learned, which I think it oftentimes is. People teach to be afraid of a snake, they teach it to their kids. But also, you're right, that it's a fear of the unknown. I mean, here's the thing. You know, cars kill 1.2 million people a year, and we don't go shooting cars or chopping the hood off the car. You know, it's a major problem, and we need to. But I'm so grateful that your teams are out there gathering the information, talking about the biodiversity and the beauty of the snakes, as well as addressing and trying to come up with ways to treat these snake bites. So without further ado, we're going to be bringing in Dr. Nick Brandehoff. Nick is he is a certified emergency medicine and medical toxicologist, and he has a passion for herpetology and specialization in research and treatment of envenomation. Excuse me. He received his doctorate at the University of California and completed his residency in UCSF Fresno. So, and he's now a fellow at the Toxicology Fellowship of the Rocky Mountain Poison and Drug Center. One last thing, he went to the University of Colorado emergency medicine, where I graduated from University of Colorado. So it's good to see a fellow buff in the house. Nick is doing incredible work with the Asclepius Snakebite Foundation and on his own. And without further ado, I'm very excited to invite Nick Brandehoff to speak and tell us about his work.
Thank you. I just wanted to say I'm faculty at the Rocky Mountain Poison Center, no longer a fellow. So I've been upgraded. So let's get started here. To my PowerPoint. All right, so my name is Nick Brandehoff. I'm going to be talking about sort of antivenom today, because I think it's quite common that people really don't understand what antivenom is. You talk about snake bites and sort of human-snake conflict. We talk about antivenom a lot, getting appropriate antivenom to the right places. But people don't actually understand what antivenom is and sort of some of the complications and difficulties in making antivenom are. No financial relationships. This is me teaching my kid how to handle a snake hook at 10 months. So when you talk about snake bites, you're really talking about 3 different syndromes here. You're talking about cytotoxicity, so local tissue damage, hemotoxicity, which is sort of some of the bleeding diathesis that you see, and then neurotoxicity, which is often sort of the either ascending or descending paralysis. The vast majority are descending paralysis, along with respiratory failure. Really, the coagulopathy and the paralysis are the main causes of death around the world. So Echis, so saw-scaled vipers, probably account for more deaths than anywhere in the world, along with Russell's vipers, and they're significantly hemotoxic. Whereas things like cobras and mambas are notoriously neurotoxic. And so being able to treat those are in areas where there's little to no medical support are important features. In the US, even with everything that we have, we still have about 3 to 5 deaths per year. The vast majority of those are people who either don't seek care late or just have really odd, rare presentations of snake bites, often intravenous injection by the snake rather than local soft tissue injury. But we have everything at our disposal, and with good medical care alone, we're able to have very minimal mortality in the US. As opposed to in our clinic in Guinea, this is me administering antivenom to a kid who had been bitten by West African gaboon viper. You can see there's really nothing there. We have our own little pulse ox that now everybody's familiar with from COVID, because a lot of people have them at home now. This was pre-COVID, but you see a little cot with no sheet there. We're using a lantern for light because the power's out. We have minimal IV supplies, minimal supportive care efforts there. So when we talk about antivenom, what exactly is antivenom, and what are we giving these patients? So antivenom is really a mixture of specific antibodies and non-specific venom components or antivenom components, along with some other proteins that may make it through the purification process. When we try to make antivenoms, we try to make it as venom specific using antibodies as possible, but there's always the leakage of non-specific antibodies and other protein fragments that make it into the mixture. So first of all, we have to start with actually getting the venom, and there's the main way to do this is to milk snakes. And so in the US, we're lucky that we have labs or zoos that are specific in milking snake venoms. They're quite skilled and technical in these scenarios. Of note, people often want to catch a snake to milk it, to sell it to a pharmaceutical company. That's not how venom is extracted nor milked. It is actually illegal to do so, at least in the US. These labs that do this are very, very regulated. Next, we take that venom that is either pooled or from a single species of snakes. So when I say pooled, I say multiple species of snakes together, and we immunize animals. So the majority of antivenoms use either horses or sheep's. There's been testing of using camels and llamas and chickens and other things, but the antivenoms that we use are, we immunize horses or sheep's from it. So crowfab, for example, which is the antivenom in the United States, uses sheep. Antivip, which is the other antivenom in the United States for pit viper envenomations, uses horses. They generate an immune response from the injection of these venoms, because venoms are basically protein components. With the generation of those immune components, the serum is then drawn from the host animals. And this process is repeated every six weeks or so with some of the animals. The timing varies based on which company is doing it. And then the serum is taken, and the IgG is then spun down and separated from the rest of the serum there. And so you get this IgG component. A full IgG is on the left. It basically has a light chain and a heavy chain, the light chain being that small piece, the heavy chain being the thicker piece there, along with that FC portion on the bottom. There are 2 binding sites to every immunoglobulin, as you can see by the red arrows there. Some antivenoms remain a whole IgG fragment. So the classic example is the Wyeth product, which was historically used in the United States. It's no longer on the market for snake bites in the US. It was a whole IgG fragment. The problem with whole IgG fragments is they tend to have higher immunogenicity, and so a higher rate of allergic reactions. And so probably most likely related to the FC portion, which is the area that binds to the mast cells and causing the mediated immune response. It can result in things like serum sickness, higher rates of anaphylaxis, things of that nature. So for a lot of antivenoms, what we do is we treat them either with papain or pepsin, which are enzymes that cleave that FC region off. And so you get either a fab fragment, which is that middle one there. You get 2 fragments, small fragments from it. That's similar to what our crowfab is, or you get a fab2 fragment, which is that right-sided immunoglobulin there. The FC portion is removed and filtered out, and so you have that same site. You have 2 binding sites per IgG still present, but the higher immunogenic portion is removed from it. And then we try to isolate those specific antibodies that we want. So we either do that through affinity purification, precipitation, a combination. But unfortunately, this process is not completely perfect, and so it's almost impossible to effectively eliminate all the specific, the non-specific components we don't want it. If we had 100% specificity rate, we would have incredibly effective antivenoms at much lower protein loads, so hopefully reducing some of the adverse reactions that we can see with some antivenoms. Some companies are much better at doing this than others, and so that's when you get into the quality of antivenoms around the world can vary heavily based on this entire chain of processing. And so you get this final product here. This is a very old antivenom that was marketed in the United States, but we've been doing this exact same process since 1894 when Calmette started this process in Vietnam. So we have a whole wide variety of different antivenoms. This is currently my research stock here. Each one is very specific to the snakes that it needs to be treating. There is some paraspecificity, and so For example, some elapids, a coral snake antivenom may have some effectiveness against some other neurotoxic snakes, depending on the venom compositions, but that's, again, highly variable. The problem we run into is when you think of snake venoms are not a simple substrate. There's over 100 different types of snake venom components. These are some of the big families of those. So you have SVMPs, so snake venom metalloproteinases, you have myotoxins, PLA2s or phospholipase A2s, which is the large family with a high variety of different clinical syndromes, phosphodiesterases, SVSPs. And so this isn't a one size fits all. You can't just take a rattlesnake, for example, and say, oh, it's a rattlesnake antivenom and have it work for all around North America, because the different species have different compositions of snake venoms. And so you may make an antivenom that is very effective against myotoxin A, for example, but you may completely miss the phospholipase A2 family if that snake doesn't have high rates of PLA2s present in their venom. So this is the example of the different venom compositions that have been documented. You can see just sort of the vast array of different venom compositions of each family of snakes here. So Echis versus Crotalus versus the Bitis species, it varies. And so this is why it makes it difficult to have a single universal antivenom. On top of that, even within the same species of snake, you can have venom variations. And so different pressures on the snake causes the venom composition to change. And so, just by evolutionary history, so we've documented well that snakes in Colorado, so the Crotalus viridis or the prairie rattlesnake, for example, the venom composition changes heavily from south to north. And that's probably to help them survive in different environments, because Colorado has a differing environment from south to north, different gene flow, different diets. And so diets in specific areas. So if there's sort of an arms race, for lack of a better term, of prey predator. So if the prey develops a resistance to a venom, there's now pressure on the snakes who have venoms that are more effective. They tend to develop higher population numbers and outcompete the other ones. Along with age, so age of the snake, the venom composition changes. So from juvenile to adults, and so it's very, very, very complicated. And from around the world, for example, for Africa, we have a couple different effective antivenoms. The problem is Africa is in a huge continent with huge biodiversity, and to say that a Western gaboon viper in Guinea has the same venom composition as a gaboon viper in Central Africa is just it would probably be inaccurate. We just don't have that data yet to look at it. And then again, with antivenoms themselves, they can vary based on variation of which venoms go into it, how much immune response occurred in the animals, which you got the snake that you injected the snake venoms to, how well you did the purification process, with some of the good stuff left in the purified agents. It's really hard to know. And then on top of this, sometimes making antivenoms can be incredibly circuitous. So for example, there's 2 spots in the US that milk rattlesnakes, four different species of rattlesnakes, 2 in Salt Lake City, 2 in Florida, to make crowfab. That venom is then shipped to the UK, where the UK certifies it. It then ships it to Australia to do the immunization part. And then they ship back the serum to the UK to be processed, and then they ship it back to the United States in order to sell crowfab. And so it's just a very long, it's a very time and intensive and expensive process. Future therapeutics, which I think Matt's going to talk a little bit about his project, but there's a few different avenues people are looking for. Antivenoms work well, but there's a lot of variation, as we discussed. There's inherent limitations such as cost, allergic reactions, getting the antivenom to the appropriate places, and then there's new developments on the way. So this is an example of a case we had in July in Guinea, just showing you the effectiveness of appropriate antivenom even in the most austere environments. This is our clinic in Guinea where this lady had been bitten by either a green mamba or probably more like forest cobra. So 2 minutes after envenomation, you can see she's still in respiratory distress here. 25 minutes after, she's now able to actually move. She's breathing better. 60 minutes, she's breathing comfortably in bed. This is me testing her cranial nerves, which are often the first to go after an envenomation and the last to return after antivenom administration. So she's looking well. And she's showing us her, everything is working well. And then this is her with her family 3 days later. She's the breadwinner of the family. So as Jordan alluded to earlier, if she goes down, then the rest of the family and their local economy ends up suffering from something as innocuous as she was hanging a lantern at night in order to be able to see as she cooked, and a snake bit her. And then this is Salou Balde. This is the director of our clinic in Guinea. And he just has a great quote that if you're getting involved in snakebite for money, you should know there's no money to be made. But if you want to make a real difference, help your people, save lives, prevent disability, and advance science, and you found the right project, which I think we're all in agreement with for us that work in this scenario. Any questions, anybody feel free to email me or call me at any point, but I appreciate your time in listening. Thank you.
That was awesome, man. Thank you so much, Nick. I mean, what's interesting is we looked at the global perspective and then we looked closer at Congo Brazzaville, and Nick, now you've given us the sort of in-depth on venom and antivenom, and wow, that does look like a convoluted system that needs upgrades, and that's what these new developments are really exciting. I'm really psyched to welcome Dr. Matt Lewin, who is Matt Lewin is a field and expedition research medicine expert. He's also a doctor of neurophysiology, California Academy of Sciences, and a fellow of the Explorers Club. So it's good to see our Explorers Club members here. And he's also he runs a company called Aphirex. I believe that's the right pronunciation. You'll tell me, Matt, but which is doing incredible work on totally new sort of ways of approaching snake bites. Very effective and in clinical trials now. And we also have Jerry Harrison here, who is here with Matt Lewin. Now, Jerry, we're going to hear about you and your interest in snakes and venom. So without further ado, I want to welcome Matthew Lewin and Jerry Harrison.
Thank you very much. Thank you for having us. Of course, Jerry is an unexpected participant. I lassoed him in the last 45 minutes to come over, but Jerry's helped me start this program, and we'll describe that momentarily. So let me get you started. So the drug that we're developing is called veraspatin, and as Nick alluded to, this is a new approach to snake bites, and I've been introduced adequately. We're a public benefit corporation that was co-founded by Jerry with me in 2015. And the way this came about was that I've been an expedition physician for the last 20 years. And while preparing for an expedition to Southeast Asia, the death of Dr. Joe Slowinski was on my mind in a really big way because antivenoms are not readily available. And long excursion times in the event of an emergency would make result in lethal envenoming, as what happened with Joe. And of course, snakebite is an occupational hazard of expeditions. In the bottom right-hand corner is a Chistrodon hallis, which is a Chinese viper that was found was sleeping next to one of my colleagues' heads in a sleeping bag while we were in the southern part of Mongolia. And this, unfortunately, in the top right corner is the crate that killed Joe Slowinski, pictured on the left. I really worried about this from the perspective of expeditions, but of course it turns out, as you've learned, that snakebite is a huge global health problem. And as I was contemplating different ways to approach this, the idea of a new approach came forward, and that was to come up with an idea to have a needle-free approach to snakebite that could be used in the field. This idea got a lot of resistance. The two groups that really recognized the potential for this, first was Jerry, who can tell you how we met, and the military. The Defense Health Agency has recently been our biggest supporter and really making a huge difference, as they so often do in tropical medicine, historically with malaria and now with snakebite, by supporting this program. But I think Jerry can best introduce how this came about. I was frustrated. Nobody wanted to hear about this idea.
So I was having a beautiful June party here where we live, both Matt and I. And I had just, for one reason or another, I'd just met a bunch of neuroscientists, and one who had become a hiking friend brought Matt. And these people were sitting around, and I went, God, there's a lot of smart people in this kitchen. I wonder if there's any great ideas for companies or for something. I go, does anyone have any really great ideas that are lying fallow? And Matt said, I do. And he started to explain to me what he about Joe Slowinski, and about the idea that something like 75% of the people never even make it to the point where they could get antivenom. And then that antivenom, that the variability of the venoms make antivenom, it has to be just the right antivenom for that particular bite. And I just said, well, we just have to do this. If you have something like this, we must find a way to bring this to the world. And we've been working on it a long time, but we're getting closer and closer.
I think that's exactly right. So as Jerry said, the variability of venom composition is really one of the challenges of the antivenom approach, as you've heard from Nick as well. And so what I think is going to happen in the future is a drug like ours, which is now in clinical trials, will act as a bridge to survival, as well as augment the antivenom technology that is really in dire need of modernization, as is snakebite treatment as a whole. As Jerry pointed out, approximately 75% of the deaths occur before victims reach the hospital. And this is what really alerted me to the fact that the idea I had, just worrying about how to take care of the expeditions, could have global health implications if it was properly developed. And Jerry said, nobody's going to take you seriously if you don't do it in a business-like way. I thought I would just publish a paper and somehow it would miraculously happen. And as it turns out, that was the easiest part of the whole project. This is a child who I saw in Tamil Nadu, was bitten by a Russell viper, which is a very deadly snake, not just because of the nature of the venom, but also because of the sheer number of bites that it causes. And just to give you an idea of how austere things can get, even in a relatively well-supplied hospital in India, this is actually the pain medicine that the child is clutching. That's what the parents can afford. And these are self-pay hospitals. And so the snakebite can be just painful and devastating, expensive, and resources are super limited. I think in the interest of time, I'm going to keep going. This slide generally summarizes on the left what Nick was discussing about antivenom, but let's talk about the potential advantages of a small molecule like we're working on as a field antidote and adjunct and complementary therapy with existing therapies as we enter this kind of hybrid era of synthetic molecules as well as traditional antivenoms. Of course, one of the big attributes that is key for us is the oral formulation. We were looking for something that didn't care what type of snake it was, that it would block the particular toxins we were looking for, and would have a low cost. And so This particular molecule that we targeted first is called PLA2. And this quite lethal component of quite lethal toxin is present in 95% of all the snake venoms around the world, and in varying proportions, as you saw in those pie charts. But the activity of this toxic component is spectacularly high, so that even venoms with relatively low concentrations of SPLA2 can be quite deadly. And what we did was we found this molecule here in yellow called verasplotib that fits in this little green area. These three amino acids are conserved in the business end of these venom toxins throughout the world. And this drug, amazingly enough, fits in the pocket and blocks them all, irrespective of geography or other structural components. And so this was a very appealing target for us. And as it turns out, it's quite effective in animal studies for blocking the venom toxicity syndromes, whether it's neurotoxins or breaking up of the red cells that cause anemia and some types of bleeding. This is an early experiment I did, just taking my own blood and mixing it with the venom of a number of quite dangerous snakes. Crate and Russell viper on the left are two of the more deadly snakes in the world. You can see that when I treated my blood, no rupture of the red blood cells, and when the red cells are not treated with the drug, you get the rupture and you get the red color, and you can just see sort of as you go across, treated, not treated. And this was just an early clue that the drug might have a desirable effect on a broad spectrum of venoms. Since we started this work, more than 20 labs have reproduced or extended our work. And now, as of today, I counted 57 different venoms have been tested from around the world and from many different labs for the effect of this drug on this lethal component of venom. So what about this drug? It has these attributes that may be appropriate as a global health solution. It's a very tiny molecule compared to antivenoms. It behaves more like an antibiotic. It can be taken orally. It can be given IV. It penetrates tissues that cannot be penetrated by larger molecules. And in this way, it could really augment the toolbox for treating snake bites in such a way that, for example, you can put it in your pocket, and if you're bitten by a snake, you would take this quite early, and you would show up and you would get to the hospital in better shape in the majority of cases, we hope. Very nice potential for children. The drug is tasteless. It goes easily into applesauce, milk, or any kind of feed, and could be taken orally. A number of recent studies have really demonstrated the compatibility with antivenom, and the fact that the combination of the two, antivenom with this drug, actually both seem to work better. And there's a paper that was just published out of Sao Paulo this week demonstrating this principle, that the effect of the two drugs together gave a superior performance for neurotoxicity, the bleeding effects, kidney damage, and liver damage. Of course, you need something that is appropriate for the conditions in which you would be using it, and this drug is quite heat stable in hot and humid tropical climates. One of the things you've heard over and over again is there's just no funding for snakebite, and one of our strategies was to look for a drug that had been previously tested. And in fact, this drug was developed by Eli Lilly for totally other reasons, and has actually been tested in more than 4,000 people over the years for conditions that never made it to market because the drug was not quite effective enough. And to that end, we've benefited greatly from this previous knowledge of the drug, and it kept our development costs quite low, with Eli Lilly having licensed us the technology of how to make it and giving us the know-how that might have cost hundreds of millions of dollars to figure out from scratch. And so we can anticipate that with the simplicity of this molecule and the fact that it's completely synthetic, that this should be a quite nice low-cost alternative compared to the cost of antivenom, and should be able to save money because of the decreased need for antivenom utilization. There are certain advantages to antivenom. For example, it has a much longer half-life. And so for severe envenomings, you can imagine having to redose this drug. But these two strategies should really improve and modernize the treatment of snakebite. This is just to show how, we were just amazed when we tested this. This was a study conducted by Dr. Yulia Surovtseva at Yale University, and comparing the potency of this drug that we're developing compared to antivenom. And these below the name veraspatab are several different antivenom brands, and you can see that it's 4 to 5 orders of magnitude more potent than antivenom. So we're now in clinical trials, and this is our current clinical trial, which is taking place. We've already enrolled patients in the United States. We're starting up in India next month, where we really hope to have the biggest impact. Early on, India accounts for half the world's burden of snake bites and snakebite deaths. And as the project grows and we mature, the technology, we really are eager to branch into African snakes, which are very complex. I really take my hat off to the groups that are working in Africa, which have a tremendously complex clinical syndromes in need of additional therapeutics. One of the recent stories you might have seen is that in COVID, the toxic components that appear to be responsible for lethality in COVID are like venom coursing through the body. And it turns out that the enzyme that we find in venom that we're targeting also appears to play a role in COVID as a result of the body's production of this enzyme. These enzymes are quite ancient. They've been invented and reinvented many times during evolution. As it turns out, the inflammatory response in humans produces this enzyme, and it behaves much the same way as venom does. And this paper came out of Dr. Chilton's lab in the University of Arizona. And in fact, we have been testing this drug in severe cases of COVID as well. And so, to illustrate this, in SARS-CoV-2 infection, as well as other inflammatory types of diseases, you can have a catastrophic PLA2 response noted here in the upper right. You get a massive inflammation and destruction of the lubricant of your lungs, and that results in respiratory failure. And it's our hypothesis that this drug can block that process when it gets out of control, or at least tone it down in such a way that you would have a better chance of success. And we've enrolled the first dozen or so patients in our ongoing trial for this purpose. So we come full circle, 2 trials, 1 drug, and maybe this is a drug that's found its purpose, which is really acute life-threatening events. And hopefully it will work for one of them, if not both. To that, I will say thank you very much. I know we've gone a little over, and I think it's time for Q&A. And thanks, Jerry, for coming over on short notice.
My pleasure.
Howie.
Thank you. Oh man, what an interesting presentation. I mean, it's really exciting to see the other side of this, the other side of the antivenom work. Not many people know, but Jerry Harrison, actually many people probably do know. He's also keyboardist and guitarist of the Talking Heads. So I'm pretty psyched to see that you two came together and made this, motivated this company. And I mean, I talk about fascinating and looks like could be an amazing combination between the antivenoms and this oral, solution. So these meds, I'm really excited to see that, and we want to know more about how the clinical trials happen. And wow, this sounds like, together with the Asclepius Snake Foundation and all the incredible field work, it sounds like there's a really exciting future. We have tons of questions, but here's what I'm going to do. I'm going to ask just one or two real questions to each person, just one question each maybe, because we're a little over time. But so if we could envision a future like with a positive future where we really make a difference in the not only for snake bites, but also with snake conservation. We want these snakes to be around. It's one of the most diverse and beautiful of all the animal kingdom. But it's these kinds of solutions that seem really to have hope. So just for the speakers that are with us, I know Dr. Kate had to run, but for the speakers who are with us, if you'd like to just say if you can talk about how you envision a future where we are really making a difference, I'd appreciate it. So I'll throw to Jordan, and then Nick, and then Matt and Jerry, and we'll call it a night. So Jordan, go ahead.
Yeah, so I think if we look at what Kate said, maybe that's part of the answer, is that the unknown component. The more effectively we're able to treat these bites, I think the more we can break away from that mysticism component, which it's often viewed as, and move it towards a medical emergency that can be handled well. So I think that we have to hit that cycle in multiple areas, but I think that certainly improving treatment is a critical starting point.
Awesome. Nick, thoughts?
Yeah, I think the collaboration of different specialties within research is incredibly important. So from Kate's end, from the herpetologic biologic conservation aspect, I think me and Matt are probably some of the few physicians that probably straddle the venomics and clinical side, and sort of trying to bring the bench research and the clinical aspect together. And then, I think the road forward as far as treatment goes, in order to help even improve conservation, is to honestly deliver just basic standard care as of right now with the effective antivenoms that we know works. For example, our clinic in Guinea, they get about 700 bites a year, so they've been able to reduce mortality from 30% to less than 5%, which is really, really basic stuff that we would consider good stuff. With the long-term road of doing stuff that Matt's doing and some other researchers are doing, where they're sort of coming up with the hybrid approach to get better field care out there, along with getting access to definitive effective care. So our greatest short term is to get funding for some of the clinics. As Kate said, in the Congo, they get a lot of bites. They have absolutely no care there. If we just got basic supplies and effective antivenom there, we can make a world of difference. And then in the long term, starting to focus on cheaper, even more effective ways of providing that care and providing that education to the people that need it most.
Thank you. Matt and Jerry,
So I think
about a positive future and a hopeful future.
I think Nick encapsulated it really nicely. We're kind of entering this hybrid era with drug and antivenom, and that aspirationally you can imagine having something completely synthetic. But I think for the near future, we're doing the right thing. I have to say, I've never enjoyed working in a field of research so much with so many friendly and collaborative people. It's by far, in my experience, been the greatest joy of my career. And I think Jerry actually was the first person really to point out to me about the possibility of using these types of drugs for veterinary care. I don't know if you.
Yeah, I mean, in fact, I think it's also one of the reasons the military has been interested in us is that the bomb sniffing dogs are often going into hiding places that a bomb could possibly have been planted, which is right where a snake has sort of taken up temporary residence, or so to speak. And so, but you could also imagine a family in India where one cow is the source of protein for the entire family. And if that cow dies, then what happens? And my vision is that what we've been developing at Ophorix is that we find ways of getting it distributed so that it can be in anyone who needs its pocket, so to speak, in the world. And then these improvements in distribution and in the quality of the antivenoms are close enough by that further treatment is right down the road, so to speak.
Oh, man.
So
That's really exciting. Matt, go ahead.
I was going to say that the hybrid to electric analogy is not mine. It's actually our CEO's, Nancy Koch, who's done an admirable job running this program, and actually was really the brainchild behind making us a public benefit corporation, so that ensured that we would be able to run the company as well as meet the mission of ensuring accessible drugs that would be accessible to the world.
Awesome. Well, I just wanted to say thank you. I've learned an insane amount of, totally novel, interesting information tonight. I mean, you all are doing cutting edge work. I'm a snake lover, and I feel like what I like about everybody on this panel is that you realize snakes aren't the problem. It's really how we treat human snake interactions. And it sounds like there's been huge, there's some really exciting aspirational, future if we can get the drugs and the antivenoms and all y'all folks working together. So I just want to say thanks again. It's cool to see a rock star and a doctor working together on a novel solution. But Nick and Jordan and Dr. Kate were just so incredible. So I want to say thank you. I've learned so much. My favorite snakes are, the one that everyone hates, which is the cottonmouth, or the AKA, the water moccasin. I mean, I love all the underdogs, all the things that people hate. But I just wanted to say thank you. This has blown my mind. I'm very excited. Keep up the amazing work. And we at the Explorers Club, pursuing science, research, and conservation. That's what tonight was all about, and human welfare. So there we go. There's the Explorers Club hat. So thank you. And I will do a little throw to next week. We have next Monday. We got an exciting night, breaking apart the Thanksgiving myth with Silver Moon Mars LaRose, and who's the director of the museum and a member of the Narragansett tribe, director of the Tomahawk Museum. You know what? We at the Explorers Club, nature, culture, history, and science. So we're excited to host next week. I'll be watching that. But everybody, again, from Asclepius Foundation, from Aphirex, all of you all, incredible evening. Thanks again. And let's continue to love the natural world and our snake cousins, but also we'll be safe and careful. So everyone else watching the Explorers Club, thanks for tuning in tonight. We appreciate your support and love for the natural world and for exploration, and we'll see you next week. Thank you, everybody.